Okay, ladies and gentlemen, you are welcome. So in the present video, we are going to look at terminologies in embryology. Don't forget to subscribe and join my YouTube community. Introduction to embryology terminologies. So first, what is embryology? Embryology is the study of the development of an embryo from fertilization to birth. So embryology is, of course, one of the branches of biology that trying to study the development of embryo from fertilization up to the point of birth. So from fertilization to embryo giving birth, that is what you call embryology. So understanding key terminologies is essential for comprehending embryonic processes. So if you really want to understand the concept of embryology, it is very important to understand the basic key terminologies in embryology. So this is actually the embryology, that is the development of embryo from fertilization up to the point of birth. And if you can see clearly from here, you can see that this is the point of the fertilization up to the point of the embryo and then giving birth. And that is what we call embryology. So in this presentation, we are going to cover fundamental embryological terms used in developmental biology. So first, what is fertilization? Fertilization is a process whereby the nucleus of sperm cell and the nucleus of the egg cells unite to form a zygote. The point you should understand at the point of fertilization is that before fertilization takes place, the nucleus of the sperm cell and egg cells must unite together then to form a zygote. That is why sometimes you can have thousands of sperm cells or millions of sperm cells. If they cannot penetrate into the egg cell and release their nucleus, so there will be no fertilization. So the nucleus of the two gamete cells, which is sperm and the egg cells, must come together. So the zygote is a single cell resulting from the fusion of gamete. So when the sperm cell and egg cells come together and they unite together, they will form a zygote. And the zygote itself is a single cell. Let's see from the image here. You can see this is the egg cell and the sperm cell. So the egg cells is contributing 23 chromosome. Also, the sperm cells is contributing another 23 chromosome, which subsequently leads to the development of zygote, that is 46 with 46 chromosome. And this zygote is a single cell. And with further development, the zygote will further develop into embryo. And of course, the embryo maintains 43 chromosome. So the point of from the point of fertilization to embryo and up to the stage of giving birth is what we call embryology. So this is what happened in the case of embryology. The egg cell and the sperm cell are contributing 23 chromosome. When the nucleus of these two cells come together, as you can see from here, then a single cell zygote will form. And from zygote to the embryo. So the zygote itself is a single cell. So the next thing, it marks the beginning of embryonic development. So once zygote is produced, so it begins the concept of embryonic development. So once you have a zygote, then the embryonic development starts. So the next thing is cleavage and blastomeres. So after the zygote is formed, a cleavage occurs. What is this cleavage? It's a series of rapid mitotic divisions of the zygote. So the zygote will cleave itself mitotically, and the resulting cleavage cells are called blastomeres, which are smaller than the zygote, than the original zygote. So in this embryo, in this zygote, the cleavage occurs, which is a series of rapid mitotic cell division, which result in the formation of blastomeres. And these blastomeres, they are smaller than the original zygote. So the cleavage leads to the formation of multicellular structure called the morula. Let's look at it. You see, this is the zygote. So the zygote cells will now cleave itself into 8-cell stitch. And from there, it will further cleave to produce a blastula. And then from blastula, it will also lead to the formation of cross sections of the blastula, which in the next step, gastrulation occur. And after the gastrulation is the invagination of the embryo, which subsequently leads to the generation of 3 germ layers: ectoderms, endoderms, and mesoderms. So this is actually the beginning of the embryonic development, where there is a rapid cell division that leads to a multicellular embryo. So it started from blastula, then to morula. From 8 cells stage, the 8-cell stage is blastula. And then 16 is morula. So this is how it starts from zygote, 8-stage cell, blastula, then the cross-section of blastula, gastrula, then the germ layers. We'll see them one after the other. So what is morula and blastocysts? The morula is a solid pool of cells resulting from early cleavage. So as a result of early cleavage of the zygote, a morula is formed. And this morula contain about 8 to 16 small cells than the original embryo. So the blastocyst is a fluid-filled structure that forms from the morula during early implantation. So you see the solid balls of the cells from the zygote is morula, and then the blastocyst is a fluid-filled structure that forms from the morula during early implantation. So then this is of course the morula, and this is the blastomere. So we have the fluid-filled component and then the solid ball component. So the solid ball component of the cells is morula, while the fluid-filled structure is the blastocyst. So the blastocyst contains an inner cell mass which is destined to become the embryo. So then the next thing is the germ layers and gastrulation. Gastrulation, what is gastrulation? Gastrulation is the process that forms 3 primary germ layers. So from the blastocyst or from the blastula, the gastrulation occurs, where the 3 primarily germ layers are developed. So the process that involves the development of 3 germ layers from the blastula is called gastrulation. And these 3 germ layers include ectoderm, mesoderm, and endoderm. So the germ layers give rise to all tissues and organs of the body because these 3 germ layers, they are stem cells, and when we say stem cells, they have the potential to differentiate into different cell types, and that is why they are called that's why they are called stem cell. Although we have different stem cells based on their ability to differentiate into cells, we have multipotent stem cell. So ectoderm, mesoderm, and endoderms are multipotent stem cells because they have the potential of a specific types of cells that develop into. So these are the germ layers, and as you can see, they are layers that can give rise to all tissues and organs of our body. So it is from them that our organs like kidney, brain, blood, and so many other organs and tissues generated in our body. So this gastrulation established the basic body plan for the embryo. So it is the gastrulation process that's trying to lay and establish the basic plant foundation for the development of the embryo. So let's see. The formation of these three germ layers is actually also occur at different intervals. So the primitive streak, which is called groove on the dorsal surface of epiblasts. As we can see, this is the epiblast here. So gastrulation occur. This gastrulation occur as a result of invagination of epiblast cells. So when the epiblast cells invaginate themselves, then there will be development of these germ layers. Like between the 14 to 15, after the development of embryo, we replace hypoblasts. As we can see, this is the hypoblast. So the hypoblast will now become ectoderm after 14 to 15 days. And then the mesoderm will develop 16 days, that is after fertilization. So a new third layer formed in between, which is known as mesoderm. And then, so you see, ectoderm are produced from hypoblasts, and then 16 days after fertilization, a mesoderm produced, which is a new third layer formed in between. And then the epiblast remaining on the surface become ectoderm. So these are the three layers. And let's look at how these three different layers differentiate themselves into different organelles. So as we can see, this is ectoderm. So ectoderm has the ability to differentiate into adrenal medulla, central and peripheral nervous system, skin, and appendages. So these are the kind of the different organs that are generated from ectoderm. And then for the mesoderm they are the fat of the stem cells, as you can see. The ectoderm is the outer layer. The mesoderm, as we can see, it is the middle layer, while the endoderms is the inner layer. So for the mesoderm can actually differentiate into kidneys and urethras, reproductive system, bone and cartilage, muscles, and vasculature, lymphatics, spleen, and adrenal cortex. So these are the different organs and tissues that can be developed from mesoderm. And then for the endoderm, GIT tract, liver, endocrine system, respiratory tract, bladder, and urethra. These are the organelles and tissues that are developed from endoderms. So they are called multipotent stem cells. Although stem cells are of different types, we have totipotent, we have pluripotent, and the multipotent. The totipotent stem cell, they have the ability and potential to produce all cell types in our body. And then for the endoderms, or these germ layers, they are multipotent. It means that they are limited. They produce or they have the ability to generate some certain cells or tissues and organs in our body. So the next is embryonic folding. Embryonic folding transforms the flat embryonic disc into a three-dimensional structure. So embryonic folding is the process that involves the transforming a flat embryonic disc into a three-dimensional structure. So this process, it involves both longitudinal and transverse folding. So the folding is crucial for forming the body axis and organ development. So that is the importance of embryonic folding. It is important in forming the body axis and then the organ development. So this is actually what it happens during the embryonic folding. So it involves both longitudinal and transverse folding. As you can see, this embryonic folding starts after 21 days of pregnancy, 23, and also 26. So you see, it involves both the uh, longitudinal and transverse folding, but that is not what we are discussing here. It's just for you to understand the importance of the embryonic folding. It's involved the transforming a flat embryonic disc into its three-dimensional structure. And it is very important for forming the body axis and organ development. So then the placenta and extra-embryonic structures. The placenta is an organ that facilitates nutrient and gaseous exchange between mother and fetus. So the placenta is an organ that connects the mother and the baby. And it's very important in facilitating the nutrient and gas exchange between the mother and the fetus. So these extracellular structures like the amnion, chorion, and yolk sac support embryonic development. So these structures develop early and are vital for embryo survival. So these are the different embryonic structures. So the extra embryonic membranes, where we have chorion, amnion, embryo, allantoins, yolk, fetal portion of placenta, maternal portion of placenta, umbilical cord, chorionic villi. So all these things we are going to discuss them in the next video. So then the next thing is embryonic period and fetal period. The embryonic period is usually spans from the fertilization to the end of the 8th week of development. So it usually occurs at the period of 8th week of development, while the fetal period is extended from the 9th week until birth and is characterized by growth and maturation. So proper terminology helps to distinguish the developmental stages in embryology. So this is actually the embryonic period, which starts from, you see, duration, first week after conception. So the major embryological event, it involves organs formed from three primary germ tissues and then emergence of basic body plan. So this is what happened. So in the embryonic period is the period where the different organs are formed from the three primary germ layers, ectoderm, mesoderm, and endoderms. So now the next thing is congenital abnormalities and developmental defects. Congenital abnormalities are the structure or functional anomalies present at birth due to developmental errors. So there are some cases where, because of the issues and the problem associated with the development of the fetus, so there are some structural abnormalities or functional abnormalities that affect the baby before giving birth, and that is what you call congenital abnormalities. So this developmental defect can also result from, so you see, the congenital abnormalities occur due to developmental errors, while the developmental defect can result from genetic, environmental, or unknown factors. So these are the factors that can cause developmental defect. So recognizing embryological terminology is essential for understanding these anomalies. So these are some of the congenital anomalies like anencephaly, anencephaly, encephalocele, closed spina bifida, open spina bifida. All these are the examples of congenital anomalies. So the next thing, ladies and gentlemen, is conclusion and importance of the terminologies. So accurate use of embryological terminology, it enhances communication among healthcare professionals. Of course, if you are able to understand these embryonic terminologies, it will help you to communicate with your colleagues. And it aids in the diagnosis, research, and treatment of developmental disorders. And the mastery of these terms is fundamental for students and practitioners in developmental biology. So, ladies and gentlemen, thank you very much for joining, and thank you. Please, if this is the first time you come to this YouTube channel, don't forget to subscribe, share my YouTube videos. See you next.